Thank you for the invitation. So it's my first participation to SBDM. I'm very happy to show some of the work. I'm taking maybe a different way to try to show you what we have been studying in the lab, trying to look at a causal action and causal way to manipulate decision making. And I will in particular talk today about using ultrasound imaging to track decision process. So if we all agree about the general question we are asking when we are talking about decision making, there is sometimes some discussion about the terminology itself. I mean, we heard about willingness to act, I mean, voluntary act, decision, and so forth, but I'm not going to go back to this possible dissociation. And instead, today I will try more to focus on what for me is also an important question is what is the appropriate level to study decision in biology? So you probably heard a lot of talking about a system, looking at the ephemeral responses or EEG responses. Some people, like myself and many others, are interested in circuit or neuronal response using electrophysiology, but some other may be interested in even at the synaptic level. And one key issue is what is the proper level to study decision in biology. And part of this question relate to what you have seen probably many times is this diagram showing the different method to study the brain and the mental state. And we have a chance, and we are lucky enough in different labs to be able to use many different type of this methodology, but they have all their advantages, but also their problematic way to study the brain. I mean, if we look at use fMRI, I mean, we will be able to map the full brain and going down in a spatial space to map a column or even large layers, but the problem will be the timing. And so the fMRI response will be quite slow, and the signal to noise ratio will be not very high, so you will need to record for a quite long session. In the other way, if you study decision making using single unit activity, you will have a very precise timing, even from the millisecond to sometimes hour. But the problem is it will be difficult to map and record, all neuron within the same or even between different brain regions. So there is a question about what we call decision, but there is also a question about how and what methodology we use to study the decision. And so what I will try to illustrate today is something that has been published or an ongoing research that I think may be interesting to causally study the way a decision is made in the brain. So the way we do that in the lab is we train monkeys to do what we call, again, based on our terminology, decision tasks. So the monkey has to look at different type of stimuli, and I will go into detail in a few minutes. And while they are doing that, we record the eye movement. When they do the task properly, they receive some reward. It could be apple juice or simple water. And while they are doing that, we can record their brain activity. So for the talk of today, I will just illustrate one very simple task I will be talking during my talk, a very task that probably many of you have seen before. So the task is mainly composed of two types of trial. In one type of trial, which call it ProSACAD, but what the monkey has to do is only very simple task, fixate a square in the center of the screen, and then there is a rectangle that appeared on one side of the screen, and what the monkey has to do is simply look at the rectangle. On the anti-saccade trial, what the monkey has to do is fixate the fixation point, but then when the rectangle appeared with the orientation telling him that he has to do an anti-saccade after training, what the monkey has to do if he wants to receive his reward is to look in the opposite direction. And of course, in this task, and I will not describe all the detail today, but we can manipulate the ratio of the height and the width of the rectangle to make it easy or difficult, and we can also manipulate the amount of reward. So what I'm going to introduce you today is what we have been working with a group of Michael Tanter for the last couple of years, is try to record the cerebral blood flow and brain activity while the monkey is doing the task. And what we described a few years ago is here. You could see on the screen that it's a monkey with a functional ultrasound probe. So here what you will observe in a few seconds is the coronal view of the cerebral blood flow recorded in the chamber of the monkey. So here the chamber was placed in the region of the supplementary eye field. Here what you could see is the right hemisphere, and here the left hemisphere. Here you can see the dorsal part of the anterior cingulate and the ventral part, and the region of interest for this first study was the region of supplementary eye field. So what I would like you to do is to pay attention to the video just for a second, just to illustrate what and why we think this is interesting for studying decision in monkey lab. Is here what the monkey has to do is wait for instruction, for an instruction to start the task. So we will record the brain activity while he's not doing anything. So here what you have on your right is the position of the eye of the animal, where he is waiting for the cue to start the task. And here what you have is in real time the modulation of cerebral blood flow. After 1 second, you see the instruction coming here, and you could see in real time the change in cerebral blood flow in the region of the supplementary eye field here. One thing to mention in this brief movie is here the value of the color bar here. It's percentage of blood flow changes. So while the monkey is start to do the task compared to the baseline, we observe some up to 50% variation of cerebral blood flow, which means that the signal to noise ratio is very high, and this will help me to introduce the rest of the talk. Okay, so now if we look a little bit closer, but this has been published already, so you can look up at the paper, is here what we did is look at the specific variation in the region of interest of the supplementary eye field. So we design the region of interest, and we look at the variation of cerebral blood flow in red when the monkey was not having any task to do, and in green when the first trial start, and with successive trial during the session. If we zoom in, what we observe is if we do a very simple detection of change in the dynamic of the cerebral blood flow, and taking 1 or 2 or 3 standard deviation to determine when the cerebral blood flow was significantly different, what we observe is that in the region of interest, so in the supplementary eye field, if we take the large region of interest here, so we cover almost the full supplementary eye field, it was extremely consistent to see in a single trial a variation of cerebral blood flow. So if we take the anterior activity in this supplementary eye field, and we look at trial before the first instruction and after the first instruction, we see a dramatic change in cerebral blood flow. As again, I want to mention that the variation went up to 50%. But even if we take only 1 pixel in the region of interest, this variation was sufficient in many sessions to determine that the monkey was in the no-rule condition versus when he started to do the task. And of course, we performed some control to verify that this is not a general change in cerebral blood flow variation. So now coming back to the main question I'd like to present to you today, and this is unpublished work, and so I'm happy to exchange even after this talk about the data. Is in some, when we've seen this very strong variation of cerebral blood flow, we thought this will give us a unique opportunity to determine some causal relationship about decision. And let me explain what we have been looking at for the last couple of months. So here again, you could see what the task what the monkey has to do. So we perform some baseline, so nothing on the screen, then it's the first trial start, and then the monkey starts to do the task, and he's receiving some reward, And all monkeys are extremely well trained, so they could perform hundreds of trials. We even have a monkey that could perform 2,000 trials in a day. But eventually, in some cases, the monkey stopped doing the task without any change. So the fixation point is still present, but it's just not doing the task anymore, and nothing happened. And eventually, in some very rare case, he is going back to the task. And so what we have been interesting to look at is compare these two single events. So the first trial, when the monkey start to be instructed to do the task, I show you a few evaluation of the variation of cerebral blood flow, but we were looking at this first response to the first trial, then the response when the monkey begin the pose, and then when the monkey start to voluntarily restart the task. So between all this time of pause, we did not instruct the monkey differently. So there is nothing we present that is instructing the monkey to restart the task. So this is really important. Of course, because we cannot force the monkey or instruct the monkey, because this is not what we wanted to do, we do not want to instruct to make a pause or to instruct to restart. In some session, this observation could be never present. So it's very rare event. And obviously now you maybe see where I'm coming to. Since we have this very high signal to noise ratio, we can really observe what is going on in the brain in this type of action, since we do not need to average across trial. So what we have been doing to look at these responses before going to the main point of the result is to obtain theoretical signal strictly linked to the instructed event. So here what you have here is the theoretical modulation induced by the trial, the instruction of the task. So here's the first trial, second trial, third, and so forth. And if you have some delay between the two because the monkey do not start the trial or immediately, then you have some delay between the two events of trial start. Then what we have done is we have computed the convolution between this theoretical signal with the hemodynamic response we extracted from each animal. And then what we obtain is some sort of theoretical cerebral blood flow signal that would be obtained if the signal recorded in the brain will match the impulse response at the beginning of each trial. So now coming to the result, here let me show you the left part of the slide first. Is here what we will be looking is the activity of a single trial, the first trial, when the monkey receives the instruction to start the task. Okay, and here we define two regions of interest. So the supplementary eye field and the anterior cingulate, the dorsal part, and in magenta and in blue it will be the activity in the supplementary eye field, and in red again this is a theoretical signal. Okay, so if we look at the comparison between theoretical signal in the supplementary eye field and between the theoretical signal, we see a perfect match. So the response of this single trial perfectly match not only the dynamic, but the time when these activities significantly change. It's more noisy, but very similar in the anterior cingulate. If we look across different single trial, for the first instructed trial, what we see is the dynamic change that match between the theoretical signal and the anterior cingulate. Now, let's look at the voluntary restart of the task. Okay, so now if we look at the same comparison, so we compare the activity in the supplementary eye field and the anterior cingulate between the theoretical signal and these two regions, what we observe is that the theoretical signal and the observed signal have a lag. And so the supplementary eye field activity restart much earlier than the theoretical signal. And it's even clearer and stronger in the anterior cingulate. So if we evaluate this lag between the theoretical signal and the physiological signal recorded in the supplementary field, but because it was much stronger in the anterior and much more robust in the anterior cingulate, what we observe is at the beginning of the task, in the unstructured condition, there is no lag between the theoretical signal and the observed signal in the anterior cingulate. In comparison, when we look at this difference between at the time of the first trial was restarted by the animal, what we observe is a very strong lag between the two signals. So the physiological signal appears earlier, for about 5 seconds before the theoretical signal. So now going back to the title of my talk, I mean, I hope I show you that we can track each unique action in awake behaving monkey using ultrasound imaging, but we are back to the question about the system. So the ultrasound, even if the spatial resolution is much better than the fMRI, I mean, still we are far from being able to look at what is happening at the cell level or even further from the synapse. So in the last couple of slides, what I'd like to show you is what we really are doing right now. It's very preliminary, is to combine these different techniques to maybe better understand the causal relationship between what the monkey is instructed to do or what is self-generated as a decision. Or, I mean, again, we can discuss about the terminology. So in our lab, when we do single unit recording, it's like ice fishing. So basically, if you've never been in a monkey or rodent lab doing electrophysiology, it looks like the right part of the cartoon. So you put an electrode in a hole and you wait to get the neuron. So you do not really see what is going on. And so you really have to of course, do a very careful penetration and wait sometimes for a very long time. And sometimes you get neuron, sometimes you do not get neuron. But this picture on the left, I mean, this is not something we have access to do extracellular electrophysiology. So what we have been developing in the last couple of months is a way to combine these two techniques. So investigating the functional cerebral blood flow and the single unit activity simultaneously. And as what you could see in the left part of the slide here is the probe, the functional ultrasound probe here that is placed particularly in the surface of the, in our case, of the parenchyma of the monkey. And here what we have is to design a new microdrive to be able to drop the electrode in the region where the activity of ultrasound is monitoring the change in cerebral blood flow. And here what you could see in the right part is a movie showing you the retraction of an electrode after the recording session. So here what you could see is the electrode is taking out and out and out and out, and you can estimate, of course, during the recording session, not only you can record a neuronal activity, but you can estimate where this neuronal activity is coming from, especially, and of course, compare this to this change in cerebral blood flow. So now I think at least I mean this is a interesting way to study a mental state at different time and spatial level, and maybe try and better understand how these cell, circuit, and system level interact. And since it's, I don't know how, if there will be another meeting next year, but I wanted to share with you some of the few recordings here of neuron simultaneously recorded with cerebral blood flow. So in red here, you can recognize the hemodynamic response we record during the task the monkey was doing. And in blue, you can recognize the raster. So each line here is a trial, and a line on when the target is presented. And here we have a nice movement-related neuron, which could be represented by the spike density function. So it's average frequency response in blue here. And we can now we'll be able to compare and combine the activity of a single neuron and compare it to the response of the cerebral blood flow. Of course, this is not something you can do by yourself alone. I mean, and I want to thank a lot all the people that have worked very, very hard in the lab to do that. So Alexandre Bizeux, Julien Claron, who just finished his PhD thesis in the lab, Fabrice Charcisset, Kevin Blaise, Serge Picot, Jean-François Aubry, and Michael Tanter. And thank you for your attention, and I will be very happy to take questions.
Thank you very much, Pierre, for this very impressive results. Okay, I may start with the first question. It's about the difference between instructed and, how do you call them, self-initiated actions or movements. So if we were in humans, there are different candidate processes at the cognitive level. So my question is about the difference between these two situations. I guess you could say something about sense of agency or taking responsibility, as Patrick Haggard would say, versus maybe some cost-benefit calculation related to motivation. Like I feel that now it's adapted to start again the task because the benefits would surpass the cost. So my question is a bit general, but what would be the cognitive process signaled by this anticipated activity you see in the SMA?
Well, yeah, I mean that's a very, very good question. So, and we are back to the question of terminology. I mean, do we and we do not have all the data yet to be able to disentangle all these different possibility or contribution, but whether it's related to attention, I think we can answer. So maybe because we could think that this is, you know, for the monkey going back to even if we have to agree on attention, but the monkey is back to pay attention to the cue, for example. But we extracted some data showing that at least in anterior cingulate, when there is only one trial, once in a while that the monkey is doing, I mean, even if it requires attention and, you know, all the saccade preparation and everything, we do not observe this change in activity in anterior cingulate. So it's only observed when the monkey is going to get back to the task for a few series of trials. So now for your question about motivation, whether that relates to motivation to restart, that's something that we'd like to manipulate. So for instance, what we currently could do, because we could have an online access to the signal, is while we detect an ongoing preparation to restart, is maybe change the cue to instruct the monkey that no, it's not going to work for, you know, that amount of reward, but, you know, to different amount of reward, and check whether with this sort of loop, whether we can extract it more precisely what could influence this activity. Another thing we try now is to, and this would be more related to what you would have liked to see in my talk, is neuromodulate. So we see a preparation, and what we are doing now is we stimulate the anterior cingulate while there is this preparation to increase this preparation activity or to decrease it, and see whether the behavior will follow our prediction. So if we decrease this preparation activity, does the monkey stop and never restart the task, or does it? Or could we make it an anticipatory restart of the task by increasing this activity? And so this is really ongoing research now. I mean, we have done a couple of sessions, so this is way too early.
Okay, thank you. Unfortunately, we have to move on because of time, but thank you again for the talk.